All right, so this is going to be a little bit of a long one. So hello everyone, welcome. So today we're going somewhere that I think is long overdue. And honestly, it's a topic that sits right at the intersection of what I do every single day in clinical practice as a wellness practitioner and what I see happening online that genuinely concerns me. So, we're talking about peptide therapy, specifically the massive gap between the real science behind these compounds and the absolute chaos of how they're actually being used by everyday people now. So here's how I want to set this up. Imagine you just bought a vial of that miracle healing compound your gym buddy swore by. He says it completely fixed his rotator cuff in 3 weeks. Sounds incredible, right? What if I told you that vial has roughly a 65% chance of being contaminated with basically indestructible bacterial shrapnel? Yeah, 65%. And I'll get to that number. It comes from peer-reviewed data published in Drug Testing and Analysis. And when I first read it, I had to put the paper down and just sit with it for a minute. Because that bacterial debris, that's really just the beginning of the problem. What we're seeing right now is a massive influx of entirely unverified, highly reactive synthetic chemicals being injected directly into human tissue. All of it based on a profound misunderstanding of what actually makes a drug safe, and those who maybe do understand, they are using it maybe a little bit better. Like myself, I've used many of these peptides, but we're going to go through the concerns around them. So let's talk about it like this. So usually when we talk about medical treatment, there's this expectation of precision, right? A lot of people feel that way. You break your arm, the x-ray shows you the jagged white line, the doctor points to it, sets the bone, casts it, and it heals. Visible, categorized. The solution is mapped out with decades of clinical data behind it. But when you step into this rapidly exploding world of peptide therapy, that x-ray machine is completely broken, basically. We're looking at something murky, unregulated, and frankly operating like the Wild West. Now, I say this as someone who has genuinely been excited about the clinical potential of certain peptides, and there are peptides that have a lot of research behind them. For example, the GLP-1s that have gotten very, very popular, Ozempic and Zepbound. Both of those are peptides. So a while back, I actually sent an email, and it was about BPC 157 research. And I was pretty giddy about the brain-gut access data. That's how excited I was. So I'm not here to tell you peptides are snake oil. The preclinical data, the animal studies, they really do show incredible scientific promise. And when I say peptides, I'm referring to just the huge category of different kinds of peptides that are out there. And we have to recognize that there are some that have a lot of evidence and they're being used and they can be prescribed and it's just a very different thing. So we're talking about, with this incredible scientific promise on a lot of these different peptides, we're talking about accelerated tissue repair, modulation of inflammation, cellular regeneration. The mechanisms are fascinating. But there is a dangerous gap between a successful study in a mouse and a safe, verified therapy in a human being. And that gap, that's where all the risk lives with these. And so, I think a lot of people, when they look at the FDA, they see this monolithic, slow-moving bureaucracy. And they're not wrong. There's a lot of issues. They see a 10 to 15-year drug development pipeline and think, that's just the government red tape keeping from healing my knee. Or dealing with a chronic illness or chronic pain. And that is actually not only incorrect in many aspects, just in the case of there being this red tape that's keeping things from coming out, but it's a common misperception. It's a common misconception in the wellness space right now. So I just want to say that there are definitely concerns, many concerns around governmental interference in healthcare, and there are definitely special interests and there are concerns when it comes to those things. But we also have to be able to differentiate between, the evidence we do have and the risks that come with the evidence we don't have. And so let me just say this, the drug development pathway does take time typically. It takes a lot of money, it takes a lot of time, but it isn't just there to create paperwork or protect monopolies. That's not the only thing it does, though in some ways it does. It exists because we do need really good data from complex, intensely difficult to work with human biological systems. And I think that's really profound. And I think we need to understand the importance of obtaining that data. So often, many of us love to think of the body as a machine, like a car. You put your oil in, gears turn better. But our body's not a machine. It's a highly complex, interconnected biochemical ecosystem. Most plants require both light and dark to meet their furthest growth potential, right? And so it is with us. Our biology responds to inputs in ways that are layered, interconnected, and often entirely surprising. Little bits of things that hurt you can actually have really profound good benefits for your body. Too much of something that hurts you does hurt you. When you introduce a novel synthetic compound into that ecosystem, the cascading effects across your nervous system, your endocrine system, your immune system, they can be completely unforeseen. So let me walk you through what this decade-long process that the FDA requires typically actually tests for, and why there are concerns around some of the fast-track stuff that's happened both in the past and currently, as well as what we're seeing, with these peptide therapies. So, and again, I just want to emphasize, some have a lot more data than others. Some are commonly prescribed in other countries. Some are very, very powerful therapies. But the vast majority lack evidence, really good evidence, and that's scary. So there's a reason why you cannot speed up the process, that you got to do it the right way. So let's talk about preclinical research. So this is just going through like our general research overview. And I won't bore you too much, but I'll go into at least the basics of it. So we have preclinical research. So this is sort of like the bench science. You have in vitro testing in petri dishes. You have in vivo testing in animal models, usually mice or rats. And this helps establish what we call basic pharmacokinetics, how the body absorbs the drug, distributes it, metabolizes it, and excretes it. It essentially asks two questions. Does this molecule have biological activity? And what is the least lethal dose in a small mammal? Incredibly important questions. But there's something to emphasize here. A mouse is not a human. Just because something works in a 20-gram rodent doesn't mean it scales perfectly to a 200-pound person. And so that brings us to phase 1 clinical trials. So these are the first in-human safety checks. So typically it's a very small cohort, usually between 20 and 100 healthy volunteers. The methodology is dose escalation. So you start with a microscopic fraction of what you think the active dose might be and slowly, carefully increase it while monitoring the subjects intensely. The primary question here is brutal but necessary. Does this compound cause immediate severe toxicity or death in a human being? It's grim, but that is the reality of phase 1. It's a pure safety and tolerability filter. And then you get to phase 2, and that expands the pool to maybe 100 to 300 people. Crucially, these are people who actually have the condition the drug is intended to treat, so that's very important. This is where we start looking for efficacy signals alongside expanded safety data. So does it actually heal the tendon? Does it lower the blood pressure? Does it modulate the immune system in the way we hypothesized? So this is where that's at. So this is where I have to play sort of the devil's advocate on behalf of every Reddit thread I've ever read, or at least most anyway, because this is the exact moment where the online forums light up. Someone posts, look, BPC 157 worked miracles in the rat studies. It didn't kill the phase 1 guys. My gym partner injected it for 6 weeks and says his shoulder pain's completely gone. If we have animal efficacy and small-scale human safety, isn't phase 3 just like an unnecessary hurdle? They don't say it exactly like that, but I think it's important to recognize that no, phase 3 trials are important. And this is what we call the ultimate filter. This is where we're looking for the rare, devastating travesties. And I think this is where it's very, very important that we understand large-scale, randomized, double-blinded, placebo-controlled trials are testing typically 1,000 to 3,000 or more diverse patients across many different clinical sites over several years. And this is the phase where the vast majority of experimental drugs fail. Why? It comes down to the law of large numbers. Human biology is incredibly diverse. We have different genetics, different epigenetic expressions, different metabolisms, different environmental exposures. I talk a lot about this in my book that I wrote recently. And so it's important to recognize that a phase 2 trial of 200 people can oftentimes completely mathematically miss an adverse event that only occurs in 1 out of every 1,000 people, right? So the sample size just isn't big enough to catch the anomaly. And that's where you have to start recognizing where this gets risky. Because if that drug bypasses phase 3 and goes straight to the commercial market and 1 million people take it, suddenly that 1 in 1,000 anomaly translates to 1,000 individual human beings experiencing a severe, possibly life-threatening reaction. Phase 3 is designed to catch slow onset liver toxicity, subtle cardiovascular failures, severe autoimmune cascades, and I want to emphasize this, autoimmune cascades that might take a year to develop. If you skip phase 3 and the most rigorous, most informative phase of human safety assessment simply doesn't exist for that compound. And so even after a drug officially receives FDA approval through the phase 4 post-market surveillance, the monitoring doesn't stop from there because there's systems in place for this. So across millions of users, physicians will then continually report data back to the regulatory bodies. And this is where the sobering reality of these peptide compounds we're talking about today comes in. Unapproved peptides, like BPC 157, injected is where I'm primarily focused here, but it still needs more data. TB 500, and most others in the wellness space, like GHK-CEU, which I'm going to be talking about later here, and there's many, many more, like many. They have largely not even completed phase 2 trials for any indication. We are flying completely blind on large-scale, long-term safety data. And I think this is really important. That said, this is where I think it's important to recognize the difference between when it is appropriate to do an experimental treatment and when it's not, and informed consent. Now, that's really important because there are people that need to do something because they've tried pretty much everything else. And if we have access to these things in a safe way, we can use them and possibly bring relief to people that are suffering. So I just want to put that out there. But that being said, this is really concerning when you think about where the peptide market currently is. So let's talk about this. Silence is not safety. While what I constantly see happening in the influencer space is that the absence of data is being falsely marketed as the presence of safety. And that's a real concern. You see these posts saying there are no documented long-term issues. Well, of course there aren't, because there's no official scientific body tracking them over the long term. You may have a single doctor out there tracking and he's got some data, but is he sharing it? There are peptide associations. Are they sharing that data? Are they compiling it? Are they actually creating safety information? Silence is not safety. That's a real concern. The most under-discussed risk here is not what we know to be harmful, it's the vast ocean of what is completely unknown. And in medicine, a void of information is precisely where the most serious systemic harm often occurs. I've been doing this long enough to know that confidence in the face of missing data is not bravery, it's recklessness. And to be very straightforward with you, a lot of people make up for the lack of data with confidence. And I just want you to hold on to that sentence for a moment. A lot of people make up for the lack of data they have with confidence. They may have personal data. They may have injected their own knee. They may have had 5 or 10 friends do really well on it. That doesn't mean that they have enough data to be influencing thousands upon thousands of people to be taking these medicines. So I think that's important. So let's go to something, and this is a long one, I know, but I'm going to go into a lot of information on here. And if you take the time, you'll learn a lot from this. I made a shorter video of this. I will share that as well. It's a little more concise, but I want to be really thorough in this. So let's go on to just some concerns, right? So the first thing is, let's talk about VEGF and BPC 157. So I bring this up because this is straight from the literature. This is an oncological risk, meaning a cancer risk, associated with BPC 157. And the reason that I bring this up is because, I don't recommend BPC 157 to cancer patients. Occasionally, the oral form and very low dose in the right fitting, post-cancer setting or even cancer setting, you have to weigh the risks and the benefits. And there are a lot of risks here, and I think that's very important to recognize. So I want to be transparent. This is where my integrative oncology background makes me particularly cautious. I've spent years training under a lot of different physicians, Gail Roberts, Charles Schwingle, Dino Prado, Donnie Yance, who's an herbalist, but also just an amazing cancer herbalist. And so I've just spent a lot of time training in the cancer world for a long time. And what I'll tell you is that I really get concerned when I understand a mechanism that could theoretically promote tumor growth. So BPC 157 is super heavily marketed for healing tissue. But what is the actual cellular mechanism driving that healing? Well, it's known to have potent pro-angiogenic mechanisms. Specifically, it heavily upregulates a signaling protein called VEGF. That's called vascular endothelial growth factor. I talk about it quite a bit. Its primary receptor is VEGFR2. And so let me translate that for just a moment. So pro-angiogenic, angio relates to blood vessels, genic means creation of, so it's literally commanding the body to build new blood vessels. If you have a torn Achilles tendon or a severely damaged muscle belly that's starved of blood flow, well, enhancing angiogenesis could sound fantastic. It can be really great. You're forcing the body to rush blood, oxygen, nutrients to the site of injury to accelerate repair. Good theory, good thought. But here's the catch, and it's a big one. Tumors cannot typically grow beyond a couple millimeters in size without their own dedicated blood supply. They need nutrients and oxygen just like healthy tissue. To get it, malignant tumors secrete their own VEGF to trick the body into building blood vessels directly into the tumor bed. So if a user has an undetected malignancy, like a little microscopic latent tumor, which, by the way, we have many of those every day as human beings, but there are other safety mechanisms in place. Let's say somewhere in the colon and breast tissue or their lungs, and then they start injecting these high doses of a synthetic pro-angiogenic peptide like BPC 157, they could theoretically, again, theory, be pouring gasoline on a fire. So giving that microscopic tumor exactly the vascular infrastructure it needs to rapidly expand and metastasize. And that is a chilling thought. You think you're healing a sports injury, but you might actively be feeding a latent cancer. Now, to be rigorous with the science, let me just be really clear here. We don't have large-scale human clinical trials confirming that BPC 157 causes tumors to grow in humans either. But the point I'm making here is that just because you understand a mechanism of how it helps someone doesn't mean that that same mechanism can't actually be causing problems in others. And that is why data matters. That's exactly why. So we have literally 0 human studies ruling it out. So it's just a blank space. And this is not a theoretical debate for the academics. For a naturopathic doctor or an integrative medicine provider whose patient population often includes cancer survivors or people who are immunocompromised, this mechanism represents a massive non-trivial concern, right? You simply cannot look a cancer survivor in the eye and confidently tell them this peptide is safe because the data doesn't support that claim. It doesn't support against it either. But the point being is that if you're saying something that just isn't true because of experiences with different patients, you just have to be aware of where the data is at. That's all. That's what it really comes down to. And the blind spot, it just doesn't shrink. It keeps getting bigger. So it doesn't just stop with oncology. There's blind spots around the cardiovascular and hormonal effects in the long term. We're talking about compounds that modulate nitric oxide production after alter vascular remodeling and interact directly with the growth hormone axis. And we have absolutely no idea what chronic manipulation of those systems does to human heart health or endocrine stability over a decade. So if you're constantly agonizing, what I mean by agonizing here is antagonizing versus agonizing, meaning you're pushing the growth hormone receptors for months on end to improve your gym recovery, you are fundamentally altering your body's natural feedback loops. The cumulative physiological toll of that alteration is completely undocumented, and that is the point I'm trying to make here. So we have a total lack of pharmacological interaction data. If you're an average person taking a daily statin for cholesterol or an SSRI for depression or an ACE inhibitor for blood pressure, and you decide to add an experimental unapproved peptide to the mix, you're operating in an entirely unmonitored chemical environment, right? So there just isn't a database that exists to tell your doctor how those synthetic peptides interact with your prescription medication in the liver CYP450 enzyme system. We don't know if the peptide accelerates the clearance of your blood pressure medication, making that useless, or if it inhibits the metabolism, causing a toxic buildup. So this really should, when you start to recognize these things, it should give you pause. And that's why I'm being so thorough in this overview. When you go online and you read these massive peptide community forums, you're looking at a fundamentally flawed data set. It's the definition of what we call the survivorship bias. It's like reading customer reviews for a flawed parachute. The only people posting are the ones whose parachutes opened, right? So take that in for a moment. The people who had a bizarre systemic immune reaction or whose latent health issues suddenly flared up, they aren't logging into Reddit to diligently update their daily peptide dosage log. They're in the emergency room or they're dealing with a complex new diagnoses. And that's what creates that particular survivorship bias. In the heavily regulated pharmaceutical world, we have deeply entrenched, legally mandated systems, and they're not perfect to catch these problems. We have MedWatch, we have REMS, the risk evaluation and mitigation strategies. These systems, yes, they're broken, but at least there's a system there. If an FDA-approved drug causes an unexpected liver injury or a severe cardiovascular event in just 1 out of 10,000 people, the treating physicians are mandated to report it. That data flows into a centralized database, gets aggregated, an investigation is triggered, and a statistical safety signal is identified. This happens. Drugs get pooled after approval down the road when we get more information. Or the FDA can issue a black box warning, right? So for unapproved experimental peptides bought off the internet, right? It's just a huge void. So if a contaminated batch of TB500 causes a bizarre autoimmune cascade or severe systemic inflammation in 50 different users scattered across the country, no one officially connects those dots a lot of the time. So just be aware of that because there's no system to connect them. The online vendor has zero legal obligation to investigate the reactions, right? Let alone report them. Other buyers are never warned. The contaminated batch just stays on the market. So you have to remember, like, you would think that human beings would say, oh, I had like 6 people get back to me saying how sick they got when they did this injection. Something must be wrong with it. And they would send out to their little group of people who maybe all went in and bought this peptide from some pharmacy in China. Well, you know, what real benefit do they get from doing that, right? If they're making money off of everyone buying this stuff, so you just have to think about those things. Like, you would hope that wouldn't happen, but the reality is oftentimes those contaminated batches can actually stay on the market. And the true incidence rate of adverse events, therefore, from these research grade peptides is completely unknown. But mathematically speaking, it's almost certainly, massively underreported. Those self-reported forum anecdotes are epidemiologically meaningless. Anecdotal evidence is pretty meaningless. You got to put it all together. So, since these compounds lack FDA approval, the mainstream highly regulated pharmaceutical supply chain won't touch them. You cannot walk into a Walgreens and pick up a vial of BPC 157, right? So that vacuum has been filled by an unregulated online black market operating out in the open, hiding behind a very specific legal loophole, the infamous research use only shield. So if you've ever browsed one of those peptide vendors websites, you'll see disclaimers plastered over every page. Not for human consumption, for in vitro research purposes only, not a drug. Now, I just want to be clear here. That doesn't mean that these things aren't effective or that they can't be, but I think we have to be crystal clear that the people running these websites are not putting that label on there because they genuinely believe you're a molecular biologist buying vials to run assays in a university petri dish. Okay? They know exactly what their customers are doing with them. It's a calculated legal dodge designed to absolve them of liability. So that dodge has really terrible tangible implications for the consumer. When a product is legally classified and sold as research use only, it means absolutely zero pharmaceutical manufacturing standards apply to its production. So no sterility testing is legally required. No potency or identity verification is mandated. No quality control documentation required by any regulatory body. No facility inspections, and absolutely no mechanism for a product recall if something goes wrong. Okay? From a strictly regulatory and chemical standpoint, when a consumer injects one of these research peptides into their abdomen, they are injecting, and I will say this, an untested, unverified laboratory chemical. Just because it's made up of amino acids that make it into a protein does not mean that it is not a laboratory chemical. And there's other things that could be in it. This is not, being rhetorical. This is not like me, this is me being literal, factual, and the legal reality of what is inside that vial. That's crazy. And so let me just give you an example of this. Like, for example, we talk about these GLP-1s, and they're a great example because they were used frequently in the peptide community while the stuff was still being researched, right? So we still had lower research data, and they turned out to be pretty incredible. They have a lot of benefits, but we also know they can have a lot of side effects. You see the big headlines where people have these really negative responses to these drugs because there are side effects with them, and they are a drug. Just because they're made up of a short chain of amino acids does not mean they are not a drug. So I just want to make that very clear. The single most acute, immediate danger of buying these is what I mentioned in the beginning. Unregulated sources isn't even the peptide. It's the endotoxin contamination. And I just want to spend a little time here to explain that. Now, I know I'm going through a lot of information. I understand. I did make a smaller video if you need to go watch that. That's fine. No stress. But if you really want to get into this, this is what this is. So endotoxins are incredibly toxic molecules. They're called lipopolysaccharides, or LPS. They're structural components of the outer cell walls of gram-negative bacteria, like E. coli or Salmonella. Um, and when you go through a complex, multi-step chemical synthesis of peptides, if the manufacturing environment isn't aggressively controlled by having an ISO-classified pharmaceutical clean room, bacterial contamination can happen. In fact, it's pretty inevitable. If you're not careful, you're going to get some bacteria in almost everything. They just get into the solution. They're in our environments. Now, your first thought might be, well, if a vendor realizes their lab isn't perfectly sterile, can't they just heat the vial, boil it, autoclave it, kill the bacteria, solve the problem? Logical thought. But there's a couple problems with that. One, you're going to maybe have some effects on the medicine that's in it. So let's just put that out there too. But heat just doesn't work that well against endotoxins. You can kill the bacteria, but the bacterial cells, that when they burst and they open and they die, they shed those cell walls, and these little LPSs, little lipopolysaccharides, these endotoxins are released into the solution, and they're very incredibly heat stable. Standard autoclaving or boiling really doesn't do much to these, and really they're going to still be there. So that's why I call them the indestructible microscopic shrapnel, right? The bacteria are dead, but the toxic debris they leave behind is floating in the vial, and you can't see it. So endotoxins are, and you're going to understand this more as I go on, why this is important for other issues. So endotoxins are completely colorless, odorless, and they're invisible to the naked eye. You can hold a vial of reconstituted peptide up to the light. It can look perfectly crystal clear, and it could be absolutely teeming with endotoxins. So what happens when a user draws that clear liquid into a syringe and then injects this microscopic bacterial shrapnel directly into their bloodstream or muscle tissue, right? The biological response is aggressive. Endotoxins are what we call highly pyrogenic, meaning they instruct the brain to spike the body temperature. They cause severe fevers, right? Because your body's saying, hey, I have a bacterial infection. I need to fight this. They trigger an acute, massive, systemic inflammatory response. And if the endotoxin load is high enough, it can precipitate what we call endotoxin shock, which looks like severe sepsis and catastrophic organ damage and even death. So, detecting endotoxins. This is really very interesting. I learned about this some years back. There was a big article, actually, that came out. I think they were informing people of this. I remember learning about it when I first learned about the horseshoe crabs, the very ancient crabs. They use these very expensive biological assays called LAL testing. I think it's limulus amoebocyte lysate testing. Anyway, this mechanism is pretty cool. So the pharmaceutical industry sort of relies on the bright blue blood of these horseshoe crabs, and their blood contains specialized immune cells called amoebocytes. Now, again, I'm bringing this could be updated. There may be new ways they do this, but as far as I know, this is still done quite a bit. These amoebocytes, over millions of years of evolution, developed a sort of defense mechanism where the blood instantly coagulates and turns into a thick gel the second it comes in contact with these endotoxins. It's like a chemical tripwire. Really cool. Really, really cool. But I'm guessing, these research use only vendors selling peptides out of uninspected warehouses aren't necessarily paying top dollar to run horseshoe crab blood assays on every batch they synthesize. They absolutely are not doing this. It's expensive, right? It requires specialized equipment. And frankly, if they ran the test, they'd have to throw away a massive portion of their inventory, which again is a cost thing. So this brings us to the most shocking statistic in all of this material. And this is what I mentioned in the beginning. A comprehensive study published in the peer-reviewed Drug Testing and Analysis systemically purchased and evaluated online peptide products. They found a staggering 65% of the online peptide products tested had endotoxin levels that far exceeded established safety thresholds for human injection. So, 65%. That's not a minor quality control deviation. That's a vast majority of the entire online market falling prey to this critical, potentially life-threatening issue. If you buy a peptide online, you have a significantly better than coin flip chance of injecting highly inflammatory bacterial debris straight into your body. Just think about that for a minute. So, and there are ways to avoid this. It's going to cost more money, and there are things you can do to sort of avoid that, right? Like, people just aren't doing it. That's the thing. Remember that they're trying to save money. So it gets worse, and this is even worse. The endotoxins are just the tip of the iceberg. So independent analytical testing of these research vials often found bacterial bioburden, meaning they actually had viable bacteria in the samples too. They found heavy metal contamination, dangerous levels of arsenic and lead. They even found visible particulate contamination, like dust and debris floating in the vials. Now, you should still look at it under the sunlight, right? You're probably going to see the dust and debris, but that is crazy. And they can be really small. You may not see them if you're not careful. So, and even if you somehow miraculously dodge the live bacteria, the heavy metals, the endotoxins, there's absolutely no guarantee that you're actually injecting the peptide you think you paid for. This is a whole other thing. The mislabeling and incorrect dosing data is staggering. Independent HPLC analysis, that's high performance liquid chromatography. Yes, I did get a chemistry degree. Consistently finds vials labeled as one specific peptide that actually contains an entirely different chemical compound. You might order BPC 157 and receive a vial containing a fertility hormone or a synthetic melanin peptide, right? Like melanotan, right? So you just have to realize that this is huge. This is a huge issue. What about dosages? Well, if a vial says 5 milligrams, can you trust that? No. You might find a vial claiming to have 5 milligrams that only contains 1.5 milligrams, right? Alternatively, and even more dangerously, it might contain 15 milligrams, right? So you could be triple dosing yourself without knowing it. There are many, way too many people have experienced dosing problems with a lot of different drugs coming from other manufacturers in other countries. So some vials contain the correct target peptide, but are heavily laced with undisclosed, entirely unknown filler compounds, right? So you have to remember that. And I looked into this actually because I thought about it at one time. I was like, you know what, I can just make these peptides and then I know what I'm getting and then I can third party test them and have more control over it. Well, it's quite expensive, first off, to do that. But there was also this side to it too that, and this is really important to just recognize, that it's not just about making the peptide. You can have a 95% or 92% ability to use these peptide machines and put all the reagents together and create, your perfect little peptide. But there's still going to be a percentage that isn't that peptide. And so you need to be able to filter and clean that too. Take out the chemicals that were used in the process. And that's a problem. So you're not just buying what you think you are. You're buying very expensive, potentially contaminated, products. And I think it's important to recognize that many of these can absolutely have zero active peptide completely. There could be nothing in them. So now you're buying really expensive, potentially contaminated mannitol powder, right? So anyway, the United States Anti-Doping Agency ran a massive investigation on this specific issue because so many athletes were getting flagged for compounds they did not take. Think about that for a minute. They found that over 20% of the black market peptide products were either grossly mislabeled or cross-contaminated with banned substances, right? Anyone can put something on their website saying, oh, this has everything in it. We do third-party testing. Here's your certificate of analysis. Okay, so we've talked a lot about these things, but I want to go into what we call the amino acid sequence problem. So this is even more, and how it affects your immune system. So let's talk a little bit about that because that's pretty interesting data. So think about this for a minute. The drug testing and analysis study that they did found that 38% of the online peptides contained incorrect amino acid sequences. Okay, 38%, right? So it's a little over a third of a chance that they have the wrong amino acid sequence in them. Let's go back a little bit. So peptides are basically fundamentally these short chains of amino acids. Like, think of them like specific colored beads strung together in a very precise, exact order. So if 38% of these products have the incorrect sequence, a bead missing or the wrong bead swapped in, what we know, it looks different, right? What does that actually mean for the body? Well, it's exponentially more dangerous than just a reduction in efficacy. So a peptide with a single incorrect missing or modified amino acid is fundamentally structurally not the same molecule anymore. Its three-dimensional shape changes. Its biological activity becomes really unpredictable. Its safety profile is null and void. But most critically, that minute sequence error can present what we call novel immunogenic epitopes to the body. This leads us directly into the concept of immunogenicity. So this is kind of deep, but I want you to understand this. I'll explain it. So the idea is that injecting these slightly flawed molecules can essentially trick your immune system into turning its weapons on your own body. Okay? So think of your immune system like this really advanced, highly sensitive facial recognition software network monitoring a high security facility, your body. Your natural endogenous proteins are like the employees. They're in the database. The camera sees their faces, recognizes them as self, and lets them go about their business, right? But if you inject a synthetic peptide that has the wrong amino acid sequence, it's like someone walking into a facility wearing a hyper-realistic mask that's just slightly off. The immune system's major histocompatibility complex, MHC, constantly scans the proteins in your bloodstream. When it encounters the poorly synthesized peptide with this modified amino acid, the facial recognition flags say this is an alien. It recognizes that the sequence doesn't belong in the database. The immune system, like the security, then mounts a defense by creating specific anti-drug antibodies to target and destroy the specific molecular shape. So the FDA's 2025 immunogenicity guidance for synthetic peptides, so this is quite literally a guidance put out there, is incredibly strict on this point. The document specifically states that even trace impurities, right? Remember I mentioned that like 90-something% amount that you get out of it. You don't get 100% pure. Modified amino acids representing less than 0.5% total substance in the vial is what they're requiring. So basically, you have to have you cannot have like really any of these. You just don't want them in the body. But even that 0.5% in that guidance they described, even that little amount can create what they call novel T cell epitopes and activate that immune response that I was talking about. So that guidance of, 0.5%, that's not a lot. So even if these are like 99.5% pure, if you have 0.5% wrong amino acid sequence peptides in there, you can actually create an autoimmune disease. That's less than 0.5%. Think about that for a minute. That can trigger an immune cascade. Now, of course, that doesn't mean it's going to just create autoimmunity right away, but we'll talk a little bit more about that. But let's talk about what happens from the lab to the consumer before we go into those things. So basically, we call this cold chain failure. And this is an issue in the peptide world. This happens everywhere. They all try to do it right, but it does happen. And not every peptide is as fragile as other peptides. Some of them can withstand different temperatures. So it's important to recognize that. And think about your own body, like protein breakdown. They talk about, we get concerned when you go above 105, 106 degrees. So just be aware of those things. So peptides are fragile, though. They can be, and they're temperature sensitive, right? The chemical bonds that hold those amino acids together are delicate. Once they're reconstituted into liquid, And even when they're still in powder form, the lyophilized powder form, they still require strict unbroken refrigeration, right? And you want to keep them sort of at that 2 to 8 degrees Celsius. So they must be aggressively protected from ultraviolet light, and they absolutely can't keep going being frozen and then unfrozen and frozen again, right? So when you buy from an unregulated vendor, you don't really know what's going on in the supply chain. When it's operating out of another country, there's zero guarantee of the cold chain integrity, right? The vial sits in scorching hot cargo plane on a tarmac and Timbuktu. It gets transferred to a freezing delivery truck, then bakes in the afternoon sun on your front porch for 5 hours. So that's something that happens. So what actually happens to the molecule? Well, the thermal stress causes severe physical and chemical degradation. It causes the peptides to form aggregates, clumps. Instead of floating freely as individual stable monomers, the damaged peptide chains start clumping together into these massive tangled molecular knots. That's crazy. Aggregated peptides have a dramatically higher immunogenic potential than stable peptides. So those giant tangled knots are basically massive red flags to your immune system's facial recognition, like I described. So the body sees them as a major threat. So that brings to the next point. So you have all of these risks, right? The manufacturing sequence, the cold chain problems, the contamination, the errors, right? They just don't happen in isolation. They can come together, and that gets really scary. So the endotoxins from that 65% contamination rate, so think about this for a minute. They don't just poison you, they actually act as like a biological adjuvant. So when we're talking about adjuvants, like for example, if you go get a vaccine, right, for something, they had to put an adjuvant in there to stimulate your immune system so that you get the proper response to the vaccine and develop immunity, right, to whatever it is they're trying to get you to develop immunity to. So that's interesting when you think of these endotoxins as being used as adjuvants, because basically anything that amplifies and hyperactivates your immune system response to an antigen is that. Now you combine that, right, these LPSs, these lipopolysaccharides, they, with the other things going on, I'm sure you can get where I'm going with this. So one of the mechanisms that happens is these LPSs bind directly to the toll-like receptors in your body, specifically TLR4. And you think of these toll-like receptors as sort of like your body's biological fire alarm system, right? When an endotoxin binds to the TLR4, it pulls the alarm, the body's instantly flooded with these pro-inflammatory cytokines, and the immune system is thrown into the state of hyper-alert, paranoid panic, searching aggressively for this invader. So let's put that all together. You inject a peptide. It has a slight amino acid sequence error from a bad lab. It got a little warm during shipping, so the molecules aggregated together. Now, normally your immune system might notice the slight anomaly, clear it out quietly, and you wouldn't feel a thing. But because the same vials also contaminated with endotoxins, now the fire alarm goes off. The immune system's already on a state of absolute militarized panic, and then the endotoxins provide the adjuvant effect, and they make a tiny, otherwise manageable structural peptide impurity massively more immunogenic than it would have been on its own. And so the immune system doesn't just quietly clear that degraded peptide, it mounts a massive aggressive systemic attack against it. And so that forms a huge army of antibodies. And then, what did you just do? You basically just pushed that plunger, into your skin, and the clinical consequences can be vast. On the mildest end, the anti-drug antibodies simply just bind to the injected peptide and they neutralize it before it reaches its target receptor, and so it just renders it ineffective, right? So you're essentially enduring the pain of the injection, which isn't much for these, for expensive, slightly toxic water. And then you move up in the severity scale, and what do you get? You get this, just like we described, a pronounced injection site reaction, massive painful welts, severe hives like urticaria, widespread all over your body, flu-like symptoms, joint pain, severe lethargy as your body fights an invisible war, right? And on the acute, like really severe end, like even worse, you could get this immune hyperactivation that triggers an immediate life-threatening anaphylaxis where your airways swell and your blood pressure crashes. So, but the thing that I worry about the most, right, with these is the insidious stuff, the stuff that the phase 3 trials would normally pick up in large groups. And that's the long-term risks. So for you end up with these outside of the immediate allergic reaction, we call this cross-reactive autoimmunity. That's what I was bringing up earlier. So the immune system becomes so highly sensitized, so aggressive towards the synthetic peptide sequence that it loses its ability to distinguish between the synthetic invader and your body's own natural endogenous proteins. And because that synthetic peptide was designed to mimic your natural peptides or proteins that your body makes, you have molecular mimicry. And the antibodies that were created to destroy the flawed synthetic BPC 157 or TB 500 start actively hunting down and attacking the structurally similar proteins in your own body that you are reliant on for your baseline health. And so you can theoretically, again, this is theoretical, but it's just helping you understand, induce a permanent debilitating autoimmune disease simply by repeatedly injecting a contaminated sequence. Okay? So this is terrible. So this makes it even worse. The standard protocols that are usually out there by the influencers and the forum users, they don't ever typically recommend a single injection. They involve daily, often multiple times a week, injections for quite a few cycles. So 8, 12, 16 weeks at a time. So every single sequential injection of that contaminated, slightly incorrect, or aggregated peptide progressively increases the immune sensitization. So you're presenting the flawed antigen and the endotoxin adjuvant to the immune system over and over again. You're literally loading the immunological gun a little more every single day, and you're waiting for the threshold where the immune tolerance finally breaks and the autoimmune cascade begins. All right, that's crazy. So, and how does this community decide how much you need to inject, right? There's no meticulous data out there to decide on dosing. So they basically take the highly rigorous science of pharmacological dosing, which usually takes a decade of clinical trials to figure out safely, and they replace it with crowdsourced guesswork on Reddit. That's crazy. So there's a phenomenon out there called dose creep. It happens when community forums attempt to establish consensus dosing guidelines completely devoid of any what we call allometric rigor. So allometric scaling is the mathematical and biological science of scaling a drug dose from one species to another. You cannot simply take the optimal dose that healed a 20-gram mouse, multiply it by the weight difference of a 200-pound human, and assume that massive dose is safe or effective. A mouse has a fundamentally different basal metabolic rate. Their heart beats 100 times a minute, hundreds of times a minute. Their surface area to volume ratio dictates entirely different drug clearance rates. They have different liver enzyme expressions and different receptor densities. Translating a mouse dose to a human dose requires complex formulas that account for these profound physiological differences. But the online communities, a lot of them are just doing simple linear math. They inject massive doses, and over time, the recommended daily dose on the forum slowly creeps higher and higher as users chase faster results or build tolerance. And they end up injecting systemic loads that far exceed anything that has ever been studied, even in a mammalian model. So, it's like asking a Facebook group how long to bake a cake, but the cake is your delicate, irreplaceable endocrine system. I just want you, in fact, do the test. See, just ask, how long do you bake a cake? See what happens. That's madness. So let's talk about the pharmacy shield. So the online black market, they have that 65% failure rate minefield, right? So let's just take that. How are legitimate licensed medical practitioners, right? Like they have these big peptide study groups. How are they, utilizing the peptide therapies right now? So there's a massive structural and regulatory difference, and it revolves entirely around the pharmacy shield. The difference between an online research vendor and a legitimate licensed compounding pharmacy is the difference between a high school chemistry set and $1 billion aerospace clean room. Okay. I was in a facility where I watched them build out these clean rooms. I saw what they looked like. I saw how they got set up. They're no joke. So we're specifically going to look at like the 503A, the 503B compounding facilities. And I just want to bring that up because it helps us understand this even deeper. So a 503A pharmacy is a traditional state licensed compounding pharmacy. Their defining legal characteristic is that they can only compound patient-specific medications based on a valid, individualized prescription from a licensed healthcare provider. No bulk sales to whoever has a credit card. They're heavily regulated and inspected by state boards of pharmacy, and that's important to recognize. These pharmacies must legally comply with incredibly rigid United States pharmacopoeia standards, specifically USP Chapter 797 for sterile compounding and USP Chapter 71 for sterility testing, right? So what does 797 actually require? Highly controlled, ISO-classified clean rooms with advanced HEPA filtration systems, constantly scrubbing the air, specified air changes per hour, rigorous multi-stage sterile gowning procedures, which I've done this before. Every single surface is relentlessly sanitized. You have this flow of things you have to do. The risk of endotoxin contamination is exponentially lower than anything coming out of an unregulated warehouse. So that's important to recognize. Every single batch of injectable medication produced also undergoes the mandatory endotoxin testing, the horseshoe crab stuff, right? And they utilize independent third-party analytical laboratories to run the HPLC testing to verify that the exact chemical identity and potency of the peptide are present. So here's the golden rule. If a clinic or vendor hands you a vial and they cannot produce a valid batch-specific certificate of analysis, a COA, from a verifiable, and I just want to point this out, verifiable third-party lab, meaning you can call the lab and say, hey, did you guys do this? Right? Proving it went through rigorous sterility and purity testing, you are playing biological roulette. So let me just say this really, really quickly. People can falsify those certificate of analysis. So just because you see they have one online does not mean that it's legitimate. And a lot of those certificate of analysis could be out of like a pharmacy in another country and they analyzed it there and they're claiming that. Or they just use the same certificate and they just change the dates. I'm telling you, this happens. So in a section 503B, the outsourcing facilities, they take it even a step further. They operate under direct, intense FDA oversight. So they're legally required to adhere to, what they call current good manufacturing practices, exhaustive documentation of every step of the manufacturing process, rigorous equipment validation, intense raw material testing, continuous environmental monitoring, regular unannounced FDA inspections, and critically, mandatory adverse event reporting to the FDA. So I just want to point this out because this is important to recognize is even with all of these things in place, we still have recalls in the regular pharmaceutical industry, right? And through the compounding pharmacies. They don't happen as much, but they definitely happen. And if you're thinking that that happens in a little manner, right? What happens when you don't have any oversight? So the legal ground shifting currently. So even with these pristine compounding pharmacies doing everything right, the legal ground is constantly shifting. The FDA severely tightened the leash on different peptides. In 2024 and 2025, the FDA released heavily revised, aggressively enforced guidance documents that restrict exactly what these pharmacies are legally permitted to synthesize. And that happened even earlier. I've been part of that for a while now. For a pharmacy to compound a substance, the bulk active pharmaceutical ingredient must either be a component of an FDA-approved drug, have an official USP monograph, which is interesting, or appear on a specific FDA list known as the Category 1 evaluation list. So, many of the popular peptides, the most popular ones, like most notably BPC 157, do not have USP monographs. are not components of approved drugs, and the FDA has explicitly classified them as Category 2 substances. That means they're heavily restricted and generally considered illegal to compound for human use. Now, the FDA has cited significant safety concerns, specifically pointing to the lack of immunogenicity data and the potential oncological risks that we discussed, right? But this is important to recognize. There's a difference between oral dosing and injectable dosing. I'm still going to go over all of these things with you. We're going to talk about the positives, what the research does say, but I'm making sure you very well understand the risks. So the agency has actively sent warning letters to the compounding pharmacies caught producing them. They're even coordinated with the DEA to seize unapproved injectable peptide products. So for a medical professional, administering or prescribing a Category 2 substance exposes their entire practice to severe medical board discipline, crippling malpractice liability, and direct federal regulatory action. So, working strictly within the legal current FDA compliant framework and staying meticulously updated on the constantly shifting Category 1 versus Category 2 lists is the absolute minimum standard of care for any practitioner today. So I do know that there have been some changes. So I just want to point that out. The peptide world is shifting, it's changing, and that there are some advocates, especially in the current space. So just be aware, like this does not apply to every single peptide. But it applies to a lot. So let's talk about the big epidemic, the influencer epidemic. I've been bringing it up. Given all of these facts, the severe unknown long-term medical risks, the 65% endotoxin contamination rate, the risk of cross-reactive autoimmunity, the FDA actively closing compounding loopholes, you'd expect this industry to be shrinking, right? But it isn't. Demand is absolutely exploding. So why is the public completely ignoring the science? And what we're really witnessing, honestly, outside of like people do see some benefits, right? Like we're witnessing a massive, highly effective influencer epidemic. A comprehensive November 2025 investigation, so not that long ago, just a few months back, by CNN, documented how this demand is being manufactured. Heavily and relentlessly driven through social media influencers, alternative wellness advocates like myself. So there are other naturopathic doctors out there who are very much advocates for it, and prominent public figures. So these groups and individuals have massive, highly engaged platforms, and they're broadcasting deeply personal anecdotal testimonials to millions of followers every single day. That relentless broadcasting creates a powerful, dangerous psychological phenomenon, the normalization of risk. So when a credible sounding, physically fit voice on a wildly popular podcast calmly describes injecting BPC 157 in their torn meniscus and waking up pain-free, the audience's perception of medical risk just plummets. We're biologically wired to trust these stories over statistics, right? We are social creatures. So we've learned to listen. When the good story's there and confidence is there, we listen. And that survivorship bias, the parachute again, pops up. The influencer isn't just posting a glossy video about fighting off an endotoxic fever in the ICU, right? They're posting a video of themselves hitting a new squat record in the gym. And the unregulated market responds accordingly. Skyrocketing interest incentivizes a flood of illicit vendors to enter the space. So to compete, they race to the bottom on price, which inevitably means what? Utilizing progressively cheaper, dirtier, and more dangerous manufacturing standards in overseas labs. So what do we actually do with all of this? In clinical practice, simply telling a patient the internet's wrong and this is dangerous just doesn't work. I've done it. It's not a good idea. You can, if you feel that's how you want to practice, do that. Patients arrive at clinics with deeply entrenched, emotionally powerful beliefs shaped by those charismatic influencers and their beliefs. And telling them no, all that does is alienate them. So dismissing them outright destroys the therapeutic alliance and drives them straight to the 65% contaminated black market. So I've always believed that healing is a partnership. I call it co-creation. not an authority structure. And that means meeting patients where they are, even when where they are somewhere I'm uncomfortable with, and that's okay. So if someone listening to this or a patient walking into a clinic is absolutely stubbornly determined to explore peptide therapy, whatever one it is, and there's some good ones, so don't get me wrong. Here is the strict evidence-grounded harm reduction framework. So here we go. I'm going to give you the numbers. Step 1, the supply chain, non-negotiable. The product must come from a licensed 503A or 503B compounding facility, period. Step 2, demand verification. You can ask for the certificate of analysis. You want the HPLC purity data. You want to know, you should have that, right? And then you can call and verify it. You want to see a peak, right, on that HPLC showing 99% or higher purity of the target peptide with no mystery peaks, right? So indicating contaminants. You must see the endotoxin levels were explicitly measured via the LAL testing. And the sterility results basically show zero bioburden, right? So you got to see this. You got to see it with your own eyes. That's where you're going to feel really comfortable with that. Step 3, stay current on the regulatory status. So if the FDA shifts a compound from category 1 to category 2, the legal and safety picture changed? Probably. I mean, the reality is there's just a lot there that goes into that. Maybe the pharmaceutical world said, hey, we like this one a lot. We don't want them making it. And they, rubbed a few elbows and passed a few bucks. Who knows? But I get it. The stuff happens. But you still have to think. If there's a safety issue, you probably need to be careful. Okay? So, step 4, screen for the absolute contraindications. So this cannot be overstated. Evaluate your individual biology or your patient's individual biology. If a patient has an active malignancy or history of cancer or an active autoimmune condition or is pregnant, that's a no. It's a stop. It's not a, oh, well, I could try this. It's a no. We don't cause harm. Sorry, I get a little frustrated with some of the things I've seen. So utilizing highly potent growth-promoting experimental peptides is an absolute non-negotiable hard stop for me. I will just share this because I had this happen with a patient of mine. Thought very highly of the patient. I wanted them to do very well. They got super into peptides. I did my best to share with them that it was, you got to be careful. This is unregulated. There's things that are good about them, things that you got to be careful about. They just were working with this random practitioner who is a peptide expert, giving a full-on, highly advanced cancer patient a bunch of different peptide types. And guess what? It didn't take care of the disease. In fact, the disease kept progressing. So utilizing highly potent, growth-promoting experimental peptides is an absolute non-negotiable hard stop. The oncologic risk of tumor vascularization and the immunogenic risk of cross-reactive autoimmunity are far too severe. Step 5. This is probably the most important one. Informed consent. Explicit, deeply thorough, informed consent. The patient, yourself, you must clearly understand and without wellness jargon that you're taking a synthetic compound that has not passed phase 3 human trials. They have to understand the long-term safety over the next decade is a total unknown, and that they're operating entirely outside this, MedWatch safety net. So the golden rule of this entire space: never under any circumstances buy, use, or recommend online research use only products. Just don't do it. Like directing a human being toward a black market with a 65% endotoxin failure rate at this point with that research out there is medically and ethically indefensible. So, closing thoughts. I'm going to bring it all together. Peptide therapy is not snake oil. It holds genuine, scientifically fascinating promise. The preclinical in vivo data is real, and the mechanisms of tissue repair are captivating. I've been excited about this science, and I remain interested in it and where it's going to go. But unapproved is not just a bureaucratic label. It is in some cases, but in a lot of cases it isn't. It represents a terrifying, cavernous lack of critical human safety data. Bypassing the FDA and buying these research chemicals online introduces massive acute biological risks. We're talking about bacterial endotoxins that survive boiling, gross amino acid sequence errors, and the very real insidious threat of triggering your own immune system to wage a permanent autoimmune war on your own body. Legitimate 503A and 503B cleanroom compounding pharmacies are the only logically and medically defensible path forward, and even they are working within a rapidly shrinking legal corridor. And I want to leave you with one more thought to sit with. If our human biology is complex enough and sensitive enough to heal itself, using these incredibly specific, powerful, naturally occurring peptide signals, what happens to our biology when we continuously, chronically flood our systems with massive doses of synthetic, unverified, exogenous versions of these signals for years on end? Are we unintentionally rewriting our own biological baselines? Are we irreversibly altering our natural immune responses and cellular growth pathways in subtle ways that might take an entire generation of epidemiological data to fully understand? You know, most plants require both light and dark to reach their furthest growth potential, and so it is with us. There's wisdom in the body's own pace of healing. There's a reason these biological systems evolved the way they did. And sometimes the most courageous thing we can do is respect that complexity is there rather than trying to override it with a syringe full of unknowns. Question your sources, demand the data, respect the sheer complexity of your biology, and most importantly, stay curious. So I hope you got something out of my very intense, way too long overview of my concerns around the peptide industry. I did a lot of research for this. I put a lot of stuff together. Go check out my new book. It's called The Code of Wellness, and I go over 9 natural laws that are talking about like the natural laws we need to do to stay healthy. I go into the 8 breakdowns of disease, and this is all great framework to help those suffering from chronic illness investigate and dive deep with their physicians, trying to figure out pathways to healing. And peptides have a place there. They do. You can use them, and they may provide benefit. But we have to recognize the risks. And lastly, and I'll say this too, artificial intelligence, it's here. It's here to stay. And I really do believe, and I talk about this at the end of my book, it's part 3, that now there's a lot of scary things about artificial intelligence. I should do a whole, thing on that. But I will say it's between the physician and the patient. And I believe it's an ethical imperative for physicians to start learning how to use artificial intelligence. There's just so much that it can bring to the table for you in terms of, knowledge and insight. But what I will say is it's not just to follow blindly. It's not to make things easier. It's to help the patient better. And so I go deeply into that, some of my favorite things there. So I hope you guys got something out of today.